This is muscle video number 16 and the fourth in our series on muscle tissues. So this will be the fourth and last video of our series on muscle tissue and the last muscle video overall. Our learning objectives for this video, first is what is a motor unit? Let's define a motor unit. Number two, we'll look at a plot and we'll describe the latent period, period of contraction, and period of relaxation and what's going on during those times. Number three, we'll describe tetany. The word tetany, sometimes you'll hear the word tetanus. And tetanus you may have heard of before, and we'll describe tetanus or tetany in a physiological sense. And lastly, we're going to look at the length-tension relationship of a sarcomere. So let's start off with the first learning objective. Let's define a motor unit. What is a motor unit? A motor unit is a neuron and all the muscle fibers that it innervates. And so a future unit, we're going to start into the nervous system. And what you really need to appreciate here is this intimacy, functional intimacy between the nervous system, there's the spinal cord, as well as the muscular system. You already know that this section right over here, if we were to zoom in, we've identified that previously, and we've called that the neuromuscular junction, where the nerve and the muscle meet. And you know all about the activities that occur at the neuromuscular junction, the release of acetylcholine, binding to chemical acetylcholine receptors, opening up of ion channels, etc. And so what should be hopefully clear by now is that you absolutely must have a neurological signal in order for a muscle to contract, because if you don't have a nerve signal, then and again, we've done this before, if you don't have a nerve signal, then you don't generate an action potential. And if you don't generate an action potential, you don't send the action potential through the sarcolemma, then nothing hits the sarcoplasmic reticulum. Calcium is never released, and therefore the myosin head binding sites stay covered over by trypomyosin, and therefore myosin can't do anything. So you're not going to see filaments slide along one another unless these binding sites are revealed, which depends on the presence of calcium, which depends on the arrival of an action potential, which depends on the propagation of an action potential, which depends on the generation of an action potential, which depends on receiving acetylcholine, which depends on a nerve signal. And so one of the things that I've often said and is that muscles themselves, there's no such thing as muscle memory. Muscle memory does not exist. It is a neurological memory. In fact, muscles themselves are slaves to the nervous system. They only do what the nervous system tells them to do. So again, what that means, if there are no acetylcholine molecules, then this muscle, for all intents and purposes, will never, ever, ever contract. In fact, it'll start to die. Now, on the way to dying, like on its route to death, it might start to kind of they call these fasciculations. You might get these little bit of like tremblings within the muscle, tiny, involuntary, small, almost meaningless contractions when your muscle starts to quiver, so to speak. And so that can happen as the muscle itself starts to die and these ion channels start falling apart, and now ions start entering in haphazardly, and there's weird electrical signals, and sarcoplasmic reticulum starts falling apart as well too. But that's not meaningful contraction. By design, this thing will stay completely 100% dormant unless the nerve itself gives it acetylcholine. And so when you consider that, then again, we should appreciate that in real life we cannot really speak about muscles and their function apart from the nervous system. It just doesn't make sense. Again, it's trying to think about the best light bulb to put inside your home without considering the wiring that's gonna go ahead and power up that light bulb. It just wouldn't make sense. So in that sense, this idea of a motor unit then should become very much second nature. So a motor unit, by definition, is a neuron. What is a neuron? A neuron is this nerve cell. It's a nerve cell. It's the functional unit of the nervous system. A lot more to discuss on neurons later in the nervous system unit. But in this particular image, we see a red neuron right over here, and we see a purple neuron right over here. And these neurons are quite long, as you can see. So what is the motor unit? Here's a motor unit with this red neuron and this muscle fiber, and this muscle fiber. Together, that's the motor unit. So neuron and all the muscle fibers it innervates. So what do we see here? We see two separate motor units, right? In here, in this slide, they've labeled them motor unit number 1, motor unit number 2. Now, if I were to ask you this question, which motor unit is bigger? Which is the larger motor unit? The way we answer that is not by measuring the diameter of the muscle fibers, not by measuring how long it is. What we mean by that is simply how many muscle fibers does it innervate per neuron. And so if you look at motor unit number 1, we see that purple neuron goes to, let's see, 2 muscle fibers. And so it's 2 muscle fibers per neuron. If you look at motor unit number 2, it's, let's see, 1, 2, 3 muscle fibers per neuron. So therefore, which is bigger? Well, the one that innervates 3 muscle fibers. So motor unit number 2 is bigger than motor unit number 1. And here's why that's meaningful. It's meaningful because in any given muscle, let's say like your biceps brachii, the motor units are all roughly the same size. Look, if one of them goes to 30 muscle fibers and the other one goes to 32 muscle fibers, really it's pretty much all the same. But what we can say is that within your body, different muscle groups, if you compare different muscle groups, the size of the motor units on average are very different. So if we look at, for example, these muscle groups, and so what I've shown you here are intrinsic muscles of the hand and intrinsic muscles of the thumb. We actually never cover these muscles in the upper extremity just because we don't really need to. They're super duper important, but I wanted to leave those off. But the gluteus maximus, of course, we've covered. It's a powerful hip extensor as well as external rotator of the hip. And so if you look at these small little muscles of the hand, let's look at this guy, flexor pollicis brevis. If you look at flexor pollicis brevis or adductor pollicis, the name pollicis tells you it moves the pollex, which is the thumb. You can imagine then that these muscles, I mean, yes, they are physically smaller than this, but what I would ask you then is that which one do you think has a larger motor unit? Meaning that if you have a single neuron, and here's my neuron coming out, a single neuron coming out over here versus a single neuron coming out over here, which one of those neurons probably goes and innervates a greater number of muscle fibers? And so pause the video and think it through. And hopefully you've spent some time thinking it through. And what you hopefully came up with was the muscles of the hand have smaller motor units than the muscles of the butt. The gluteus maximus has really large motor units. It's on the order of something like a couple of hundred muscle fibers per neuron coming down, whereas this one will come down and it'll go to just a handful, maybe 8 to 10 muscle fibers per neuron coming down. So these muscle units are much larger. So that I don't want that to be something you memorize, but something that it makes sense to you. So if we think of it this way, if you consider going into a large lecture hall at some school where you have one professor and 500 students versus a smaller classroom where there's one professor and 10 students, in which of those environments do you think you'll have greater flexibility if you need to make an adjustment to the course schedule, adjusting the syllabus, changing the expectations, increasing the pace, whatever that might be, whatever adjustment that needs to be made? Where is it easier to make a quick adjustment? It's going to be in the smaller classroom compared to the classroom of 500 students. So a 500 to 1 ratio is quite different from a 10 to 1 ratio. So similarly, when you take your hands and you do very complex fine motor skills, whether you're typing or whether you're moving a scalpel because you're about to perform surgery on somebody, or whether you're typing or playing a guitar or whatever it might be, these fine motor skills require a lot of precision from the nervous system where you want to have really nice tight control over these muscles. Whereas this one, there really is no fine motor skill. It's really gross motor patterns. So nobody is going to do surgery with I'm not trying to be crass here, but nobody's going to do surgery with butt muscles. They just don't have the level of motor control that you would with your hand. And so that's where the concept of motor unit size becomes really important. Okay, so now you know the definition of a motor unit, and that was our first learning objective. So now let's go into this next concept of latent period, period of contraction, period of relaxation. This actually, there pretty much is some intuition for you guys already. What we have here is a nerve signal. See that red arrow right over here? This red arrow is saying the nerve goes zap. Okay, so the nerves go zap, and let's just pretend it dumps off, whatever, a sufficient number of acetylcholine to generate an action potential. And we get one action potential generated. What we see here is a latent period. And what we mean by latent period is that there's a period of time, this is time over on the x-axis, there's a period of time where nothing happens after you stimulate the muscle. Well, why is that the case? Well, before we explain that, let's look at what happens. So there's a short window of time, and if you had to look here, I don't know, what is that, 3 milliseconds or so? We don't need to memorize the numbers here, but maybe 2 milliseconds, maybe 3 milliseconds, whatever it is. And then when the muscle starts contracting, on the y-axis is percentage of maximum tension possible by that muscle. And you'll see at the beginning, the muscle generates very little tension. And as you continue to go along in time, the muscle generates more and more and more tension. and then it peaks, and that is the period of contraction. From there, the muscle starts relaxing, and it goes all the way down over this window of time following a single stimulus. And the fact that it's a single stimulus here is really, really important. Well, so what is it that accounts for this latent period and period of contraction and period of relaxation? Well, it's stuff you already know. And so look, this is my single stimulus, zap. The moment I zap the motor end plate, do I see muscle contraction immediately? I don't. It takes some time. The action potential needs to travel down the sarcolemma, needs to go down the tibial, needs to slap the sarcoplasmic reticulum, calcium needs to be released, and from there calcium needs to diffuse off to troponin. And all that takes at least 3 milliseconds, okay, according to that image that I just showed you. So that, in part, then, is going to explain why we have that latent period, why we're not seeing an immediate effect after the contraction. And so, again, that's that latent period. So now let's, did I say after the contraction? I meant latent period before the contraction. That's what I meant. So now, if you look at the period of contraction, why do we basically ramp up tension in the muscle as opposed to immediately hit the peak? Well, the reason for that is this. Look, these are calcium ions diffusing out. And let's say you got a whole bunch of sarcomeres just here, just waiting in line. Well, what ends up happening is that these calcium ions are at the front of the herd, so to speak, and these are at the back, and there's more coming out. And so these calcium ions are going to arrive first. So at the very beginning, for a brief moment in time, I've got 5 calcium ions bound to troponin, different troponin molecules. And then a snapshot later, I've got 10 calcium ions bound. A snapshot later, I've got 15. A snapshot later, a millisecond later, I've got 20. And a millisecond later, I've got 50 of them. Because, the first one out of the gates is the first one to arrive onto troponin. And so for that reason, what we're going to see is a ramping up period during that period of contraction. That's why it takes a little time. Now, at this point, again, all the calciums that are going to be released, let me just make up a number. 1 action potential gets sent through, and I spill out 100 calcium ions, okay? So in the beginning, let's say right over here, only 5 of them have bound, now 10 have bound, now 20 have bound, now 30 have bound, now 40 have bound to troponin, now 50, now 60, now 70, so on and so forth, right? So now what is it that accounts for the period of relaxation? Well, if you remember that sliding filament model, the way we described it is that calcium gets released, but what ends up happening is that calcium is going to get vacuumed right back up into the SR. So there's only a really brief moment in time where there's 100 calcium free floating in here, because just a millisecond later it's 99, then it's 98, then it's 97, 96, 95, 94, 93, all the way through. And so little by little, all this calcium then starts traveling back. It gets sucked back up into the sarcoplasmic reticulum. And you know that calcium binding to troponin is what allows, through moving tropomyosin, is what allows myosin to bind to the actin filament. So for that reason, this is why we see that period of relaxation, because right now I have 100 calciums bound to 100 different troponins. Now 99 of them are bound because that one got sucked back up. Now 98, now 97, now 96, now 95, now 94, 93, 92. I guess I should have made bigger gaps here. 30, 20, 10, 9, 8, 7, whatever. And so this is why that period of relaxation occurs like that, because calciums are getting vacuumed back into the sarcoplasmic reticulum. Okay, so there's our latent period, period of contraction, period of relaxation. And now you know what explains these things. What you'll see over on this slide is just, you'll see that that pattern is a little bit different for different muscle groups. So extraocular muscles, these are the muscles that move the eyeballs. You'll see that the shape of this thing is quite different. It's straight up and straight down. Gastrocnemius takes a little bit longer. Soleus takes a little bit longer. And all of this is simply because of the different properties for how long it takes or how long it takes for calcium to diffuse out, or more likely is the efficiency of these calcium ATP pumps in sucking back up calcium. So we don't really care about this. I don't care that you know the different shapes between different muscle groups. You just need to be aware that that is the case. All right. So here's the question then. Let's say you want to have tetany. Tetany is sustained contraction. Again, tetany or tetanus is the same thing. Now, this is not the disease tetanus that you might get by stepping on a rusty nail, for example. This is physiological tetanus. And all it means is sustained contraction. Now, this is a good, desirable thing. If you have a cup of coffee and you lift it up off the table and you're holding it not to your mouth, but let's say just right in front of your chest, then you want to be able to hold that cup of coffee there. Your muscles want to be able to hold on to that with sustained contraction. Your biceps wants to keep your elbow flexed in sustained contraction. Well, without dropping it. And so you want to be able to have this tension at a constant rate, just like this, okay, just like this. So how do we do that? Well, the way we do that, if you look at these S's, S means stimulus. I need to keep stimulating my muscle. So I stimulate it here, and then this is what I see upon one stimulus. And that should look a lot like this, something that we're already familiar with. Now, what I want to do then is that if I want to now start to get towards tetany, if I stimulate it twice, look what happens. I stimulate it, I start contracting my muscle, and as it starts to slow down, as I start vacuuming calciums back in, I stimulate it one more time, and I release some more calcium. To say it differently, I've released 100 calciums, I'm vacuuming them back up, and by the time I get to like 75, 74, 73, 72, I stimulate the muscle again, spilling out another handful of calciums. And so now I'm up to 150 or 180 calciums, let's say. Imagine what happens, 180, 170, 160, I get vacuumed back in, 150, 140, and now another action potential arrives, and now I spill out even more calcium. That's what we're seeing in this type of scenario right over here. And it just so happens that if you continue to repeatedly stimulate the muscle, if you continue to repeatedly stimulate the sarcoplasmic reticulum to constantly spill out calcium, then now there's perpetually calcium available here. Troponin keeps the position it's in because calcium is bound to him, and so tropomyosin is slid off the actin molecules, and therefore the muscle continues to contract. So this right over here is sustained contraction because I kept stimulating the muscle. Okay, I thought I could get this all done in one video, but just to make it not too long, I'm going to break this up, and I will continue with this in a follow-up video.